Hi, my name is Lauren Subramaniam. I am an NCSSM Durham residential student in the Extended Mentorship Program, and my research this summer was focused on the genetic effects of using Wolbachia to reduce the spread of dengue from mosquitoes to humans. Picture this. It's a hot, sunny summer day, and you just came in from being outdoors. You're fixing yourself a cool glass of water when suddenly you begin to feel it, the dreaded itch of a mosquito bite. You look down at your arm and see the pesky rash beginning to form, knowing that for the next few days, you're going to be dealing with the annoyance of that bite itching all the time. However, in countries like the US, we're lucky that for most of the time, that pesky mosquito bite isn't going to cause much harm, other than the annoying itching. For other countries across the world, over 100 to be exact, a simple mosquito bite infects over 400 million people each year with the dengue virus. When dengue becomes severe, it is often deadly, especially to children. Countries which deal with this virus typically use large amounts of pesticide to control the virus. However, these chemicals are both ineffective and harmful to both people and other organisms. This is where my research comes in. Instead of using pesticides to control the spread of dengue, scientists are using another method, Wolbachia. Wolbachia is an endosymbiotic bacteria. This means that it forms relationships with the insects that it infects. When Wolbachia infects mosquitoes, it changes their reproductive systems so that males which have Wolbachia can only mate successfully with female mosquitoes which also carry the same strain of Wolbachia. This means that over time, Wolbachia will spread through a mosquito population till all the mosquitoes have Wolbachia. This matters because mosquitoes which carry Wolbachia have a harder time transmitting dengue to people. With the help of my mentor, Dr. Jennifer Baltziger, I am looking at the population of effects of releasing mosquitoes which carry Wolbachia in Ecuador's Peru, which is in an area of the world where dengue is a major problem. We're proposing to look at how the release of Wolbachia by other researchers changes the genetics of the mosquito population in Ecuador. To do this, we're using mitochondrial DNA, or mtDNA for short. When you hear mitochondria, you may have flashbacks to your 6th grade class saying that mitochondria is the powerhouse of the cell. And while this is true, mitochondria also plays other important roles. Specifically, mitochondria carries its own genome, which does not experience as many mutations and changes as regular DNA. We can use mtDNA to see how Wolbachia changes the genetics of the Ecuador's mosquito population, because Wolbachia and mitochondria get passed the same way. My research is planning to look at the frequency of the mtDNA haplotypes, that is, the different sequences or patterns within the mitochondrial genome. We plan to see if the patterns of haplotypes change in the population before and after the release of Wolbachia by sequencing the mtDNA in the lab. My research specifically will be genotyping the mosquitoes before the release of Wolbachia to get a baseline understanding of the types of haplotypes which exist in the population. We will also be testing for the presence of Wolbachia initially within the population to see how many of the mosquitoes in the population carry Wolbachia before the release. When this research is finished, we will be able to see how exactly Wolbachia affects the genetics of the mosquitoes so we can release Wolbachia in other places in the future. Our hope is that in the future, with the help of Wolbachia, the spread of dengue will begin to slow until that pesky mosquito bite will be just that, a pesky mosquito bite.